because development is everything. You know,
That's
what
correct.
you just described, synapses and memory, this is also development,
Yeah,
you know?
correct. Totally
So
correct.
development is everywhere. Some people even say that neurodegeneration is neurodevelopment, in fact, right? Aging is neurodevelopment. So, I mean, you really have to make sure what you're defining. In my case, it is the OG embryonic development of the brain, you know?
Yeah,
Like,
this is
so
very
that's
distinct.
very distinct, yeah.
Hello, everyone, and welcome back to the Neuroscience and Beyond podcast. I'm your host, Willem, and together with my amazing team of graduate students, we are on a mission to bring the wonders of science, especially neuroscience, closer to you. Today, we have the pleasure of hosting Dr. Mateusz Ambrozkiewicz. He leads a lab at the Institute for Cell Biology and Neurobiology at Charité in Berlin, one of Europe's largest university hospitals. His lab studies how protein synthesis and degradation shape neuronal circuit assembly in a key brain region called the cortex, using advanced genetic tools and interdisciplinary approaches. His research sheds light on brain development and neurological disorders, including autism spectrum disorder and intellectual disability. As a member of the prestigious FENS-Kavli Network of Excellence, Dr. Ambrożkiewicz is also helping to shape the future of neuroscience. Tune in to discover the fundamental principles of brain development and learn more about organizations advancing this exciting field. Before we begin, I'd like to mention that this podcast is a separate activity from my position at the University Medical Center Göttingen in Germany. This podcast is supported by the International Max Planck Research School for Neurosciences, the Cluster of Excellence Multiscale Bioimaging, the European Neuroscience Institute, and SFB 1286 in Göttingen. If you'd like to support us, please subscribe to our channel and do not hesitate to leave us a comment below our content. And now, dear listeners, please enjoy the conversation with Dr. Mateusz Ambrożkiewicz. Hello, Dr. Ambrożkiewicz.
Hi.
Thank you for coming all the way from Berlin to Göttingen.
Yeah, it's not so far away. Thank you for having me. It's a great pleasure to be here, especially having seen all these incredible stars that you have invited to the podcast.
Come on, you're the next one.
Okay, next one to come. Okay.
The next shining star. So, guys, I'd like to share with you a story about how we met. It's a funny one. So, roughly 4 years ago, I used to live in Berlin, and with a friend of mine, we went to one of the lakes in West Berlin, the western part of the city. And I met Mateusz. So he started asking me, what are you doing after your university? Where are you going? And I was very excited to tell him that I was accepted into one of these graduate schools in Göttingen as well. And he looked at me like a little bit strange and started asking me these weird questions like,
I did.
Which program? Is this the neuroscience program?
Yeah.
And then he said, where are you going to live? And I'm like, what is this guy asking me, you know?
What are these questions?
And it turned out that he was also part of the program, and we used to live in the very same apartment.
Yes.
How crazy is that?
Very same dorm. And I mean, there's not a lot of, there are the two programs in Göttingen that I think are, quite favorable, I would say, among students. So the International Max Planck Research Schools. So if you hear master's program in Göttingen, you can go either, molecular biology or neuroscience. And then it just, I don't know, it kind of is one of these moments where you think the world is really small. And we had a common friend too that was also wild. And I mean, Berlin is not a place where you just randomly meet people that share similar stories as you do. Right?
Exactly, it's a huge place. Yeah, but it was like such a memorable. And now for the people who are not seeing the guy behind the camera, he's also living in the very same apartment.
So that dorm is that. Listen, that dorm has seen like a lot of a lot of neuroscientists that pass through that dorm room.
Amazing. How did you feel when you were a part of this program?
The program, oh my God, coming to Berlin and Göttingen to begin with is not a far distance, right? It's 2.5 hours with the train. However, I don't know, I haven't really visited after I've left my PhD home. And now to be back, it is very nostalgic. So you walk these streets and then you see all these amazing Max Planck buildings and the physics department, and you can also appreciate how much the city expanded in the direction of science. So now the Max Planck for Sonnenforschung, that was not there when we were here, and you guys have like really cool mensas now too, which is nice. The program really was it was life-changing for me, for sure. I was at that point I was in Poland, and I finished my degree in biotechnology, and I didn't really know what I was doing. Apart from that, I knew that I wanted to be a scientist, and options in Poland at the time were limited, but I just really wanted to explore, and I had this craving for something challenging, and I've applied, and I remember that I couldn't believe that I got in. I mean, even at the interviews, the people that I've met, it's my family. It's my second family. They became really, really close friends. We still are in touch. And so the program, of course, educationally, it also was life-changing in a way, right? Because it exposed me to the realm of Max Planck, to incredible scientists along the way, also friends and colleagues. But I have to say the friendships that you form during that time, because it's tough, right? You have to study a lot. These are really lifelong, and I'm sure that for you is similar too.
I mean, in my case, it was a little bit different because,
Okay.
We came during COVID time.
Oh, okay.
So
That
basically,
is,
the social part of the program was killed.
Right.
We had to stay at home, had all the lectures online, or most of them.
Yeah.
And however, we found a way to, interact with each other, going
Sure.
outside, etc., etc. And indeed, there are some friendships that will last for quite a long time. But yeah, it's a really intense place to be. And basically, we had lectures and studies from 8 in the morning till 6, 7 in the evening. And then you continue studying Saturday, Sunday, etc. Yeah. It was really intense, but I think it's worth it for sure. And fast forward now, 2024. You're already a group leader.
Yes.
Currently at Charité.
Yes, currently hosted
in
at the
Berlin.
Charité. Yep.
What do you do?
Right. Where do we start? So we indeed, I've indeed managed to. It is all, still sometimes I pinch myself because this is, in a way it is unbelievable. My group is at the Charité currently. It's a small team, but we value atmosphere and people really. So we invest into people as much as we can. We look at, you mean scientifically what we do? Scientifically what we do, we are interested in how to build a brain, if you will. We are interested in the molecular and cellular sources of diversity of cell types in the cerebral cortex, which is the outermost structure in our brain. People call it the evolutionary achievement of mammalian development, right? People also call it a biological substrate of cognition, and so we look at very early events that happen in neural stem cells that determine the outcomes in terms of the types of neurons that are then generated from these cells. Mm-hmm. Essentially, that's what we do. But there's so much more to it because we also are involved a lot in heavily in teaching in the medical faculty. We do a lot of extracurricular stuff, but I guess we can get into it in the course of that talk. And so that's essentially that's what we do.
During the last retreat, I remember you mentioned that you are very happy to be able to teach anatomy of the neck in the medical student in German.
Very happy, extremely. No, this is kind of the system, right? So there is a payoff. You can do research in I think a lot of universities have shared that system where researchers are also actually primarily supposed to give back, right, and teach the curriculum. And we are hosted by the Department of Cell Biology and Anatomy. So we are involved in histology teaching and anatomy teaching, but I'm not an anatomist, so this was also a big challenge for me, also to do it in German. But I've had incredible help as well and mentorship from senior professor Gabriele Rune, for example, or people that are around. They are always very, very eager to help, and this was terrifying, but it's great now. It's exciting. Yeah.
Challenging, but now
Challenging. I mean, things that are terrifying, I feel like they turn out to be exciting also at some point.
Yeah.
Yes.
So, okay, let's go back to your research topic.
Yes.
You dropped some terms:
I
stem
did.
cells, different cells, cortex, etc., etc. For people who are not in this field, we have to really make it clear that it's extremely hard to study this part of the neurodevelopment because a lot of it happens in utero already, meaning
Correct.
when the fetus is in the uterus of the mother, so we do not have access to that. How do you approach these problems?
That's a good question. And I think that indeed what makes this research exciting is that it is challenging. So you have to find creative ways of addressing it. But I think we have nailed it down. And again, I haven't done it myself. This is mostly because of the incredible people that I've been exposed to and my team now that is still, doing these things daily. I think maybe what we could start with is saying that we work on mice. So we use mice as a model, and we're using a kind of a wild-type control mice, but also genetically modified organisms to model certain genetic scenarios, if you will. And how do we approach the fact that it's challenging? First of all, if you compare the cortex in humans and in mice, then you see, obviously, you see a dramatic expansion of the surface area, right? It's crazy how many neurons populate human cortex. Also makes sense. But if you look at the thickness of the cortical plate, what we say, so the layers that are so famous. So cortex is composed of 6 layers, each is populated by a distinct type of neurons and other cell types. This did not really expand much, so it's 1.5-fold change, and this would indicate that there are some really very tightly conserved evolutionary processes that are operating in both mouse and humans. And that's why we can use mouse to understand a little bit about a principle that also orchestrates the development of a human cortex. Of course, there are differences, right? And the human brain and human cortex is very distinct. And these processes nowadays we are starting to understand because of the organoids, which we can also talk about in a future. But most of our work is concerned with animal models, with mice. And we like to take time, and we like to address things in vivo. And this is because a lot of development happens. It doesn't, people use cultures, people use different in vitro approaches to model development. But we do believe that in vivo really allows you to study systemically the process. So we use a lot of in vivo DNA manipulation techniques like in vitro electroporation. You can also use stereotactic injections. You can, so there are ways to do this. Of course, in a controlled, ethically approved by committees manner. So our lab really kind of builds on in vitro electroporation technique to address a lot of these genetic scenarios and molecular contexts that we study.
This is one of the fancy methods that you can introduce genetic material in the uterus while the fetus is still developing, and then study the development of this kind of like gene expression or the phenotype of the animal, etc.
Yeah, so correct. So I was just recently giving a talk to one of these events in the Berlin Science Week, and it was in front of the Naturkunde Museum, and it's one of these events where they ask scientists to stand on boxes. It's kind of like a speaker's corners, and we would have to explain, what we do. To people that would pass by, the passersby, basically. And I thought about, how do you explain what you just said to a person that wants to kind of understand what, the taxpayer money goes to, which I think is very important. And so I kind of compared it to like taking a photo of a family after 1 year, after 2 years, after 3 years, after 4 years, and looking at what happens to the kids. So if you think about a stem cell, it is, the source of the lineage, what we say, or the family of cells that are produced at a given time, at a given space in the brain. And by in vitro electroporation, you can actually ask a question: what does the stem cell differentiate into over the course of development, right? So it is very, very useful in terms of looking at diversity of cell types in the cortex. Yeah, exactly.
So when you say stem cells, so this is like let's say that this is the 0 patient, okay, and then out of this 0 patient you have a lot others that emerge out of it. If you wish the spread of the disease, right?
Okay.
Yeah. For example, this is just metaphorically speaking.
We love a metaphor.
Not at all like in the reality,
that's
right?
Fine.
How do these stem cells know what should be a neuron, what should be non-neuronal cells, right? In the brain, we have the so-called glia, or they're very interesting cells. There are mainly 3 types: the astrocytes, the oligodendrocytes, and the microglia. And of course, we have some blood cells, blood vessels, and cells that support these blood vessels, etc. But let's put it that way: how do stem cells know? Okay, I will become a neuron, or I will become a glia.
Well, that's essentially what my lab is trying to figure out. So, and not only my lab. So there's many labs in the world really who try to address this exact question. So yeah, I mean, if we manage, then we can meet maybe in a couple of years for another kind of discussion, But essentially, the history of the discoveries actually that were made already from the 70s onwards are, the story of that is really fascinating. But we still don't have the full picture. There has been quite gravitational technological progress in the last 10 years for sure. For example, with the single cell transcriptomic approaches, we really now can define and understand a lot of these things that happen in these stem cells and also in immature neurons and glia, like you mentioned. But I think what is for me very fascinating are the first experiments that were done, among others by Magdalena Götz, for example, who is now in Munich, who showed that actually the source of stem cell. The source of neurons, the cells that are the source of neurons actually have glial characteristics, actually per se. And that what is also quite fascinating is that the stem cells are programmed to give different offspring in a very defined, tight developmental windows. So when the embryo starts developing and there's at some point what we call the neural tube and it closes, and this is the primordium of our brain / cortex. And right at the beginning, these stem cells generate neurons that allow the cortex to connect with structures outside of the skull. So for example, with the spinal cord, we call them the deep layer neurons or layer 5-6 neurons. And then the stem cells, once these layers are established, they move on to generate layers that are positioned right on top of these deep layer neurons and establish connections within the cortex, for example, between layer 2-3 neurons. And then so what's fascinating is that same kind of niche and pool of stem cells, once the neurogenesis is complete, is now making glia. So really and then also as oligodendrocytes, they're not the same cells, right? It's not just one cell that just kind of sprays it all out there, but it's that niche. And people have been trying to understand, there are certain molecular scenarios where you can program them into, for example, making them into deeper layers, making them into upper layers, but I think we don't have a recipe yet. We know how to turn them towards a certain fate, right? But we cannot program the stem cell in a way that now is just going to make upper layers, and then that's it. So there's always some sort of a developmental consequence of that.
Yeah, we still learn actually, right? Because now we know some of the recipe, and this is how we do the organoids, which are basically we studied this programs of the stem cells, and then we can say, okay, if I give you this and this and this amount of molecule A and this amount of molecule B, you can be maybe a neuron that excites other neurons, right? Or a neuron that inhibits other neurons. But still we don't know. These are the radioglia, right? That are
so the terminologies. This field is really terrifying. Like if you just if you're just like hanging out in a classroom and you're thinking about neuroscience and you pick up a developmental neuroscience book, that was written in the 90s, it's terrifying. And I remember that also because we were doing, these mnemonics when we were studying for exams with my dear friend Steve, who was also here studying with me from Norway. We kind of looked at these genetic programs that were listed in these books, and we looked at each other and we said, it's impossible. There's no way, Also because the early data that came was really using a lot of mutagenesis, they were doing the chemical mutagenesis, and then the gene names are really derived from some weird abbreviations. It really doesn't make much sense. And then once you kind of overcome that first initial hurdle of trying to, build it in your head somehow, you kind of start understanding that behind these genetics, there's cellular phenomena that happen. And then there's different terminologies for different stem cells. Wait, I call it stem cells, but it's just really the niche of progenitors that have a certain potency, either to generate the entire neural lineage or to be more restricted. And as development progresses, obviously they are becoming more restricted, so they change their multipotency to Exactly. So the radial glia is just, first that people talk about neuroepithelial cells. And these are the OG stem cells that symmetrically divide to recover their own pool. Now, radial glial cells in a mouse, a human, also have different capacity to be able to already generate neurons. So they're also a step further towards, what we say, their commitment.
It's super complicated.
It's complicated, but actually, if you really think about it, it already makes sense because it's quite intuitive, right? You cannot be doing everything. You probably do a lot when you're a student, right? You go to do sports, you go to do your hobbies, you're creative. But as you go in life, you stop having time for a lot of things, so you're more committed towards like one task. And it's kind of similar to what happens in the embryonic murine stem cell niche, right?
That's a cool analogy. And to be very scientifically precise, and since I'm a alumni of Kettenmann Lab, microglia, they migrate from different places.
Correct. Right. So microglia come from the yolk sac now, and then they come in a cortex from a different source. So this is also, this kind of systemic interplay of cellular types, and this is kind of why my lab is really focused on in vivo studies, just because if you do this in an in vitro system, you miss a lot of these factors that you just cannot model, right?
Exactly, that's correct. So in vitro, just to clarify it, in vitro are mostly like cultures that, for example, let's take a mouse, hippocampal neurons or neurons that are derived from one region of the mouse. We have a brain that we dissociate into cell types, and we grow these neurons from the hippocampus in a dish, plastic dish with certain medium, so that the cells can grow and be happy. And this is like for 2, 3, 4 weeks maximum, and we can study them in the dish. That's why this is what is in vitro. Then you have also slices that you can, basically brain slices that you can have. The organoids are a very interesting system we can touch upon a little bit later. But in vivo means when you study certain phenomenon in the living animal. You can do it by, as you said, these genetic manipulations. You have the in utero, you have CRISPR, you have other techniques. You can also image live. We already had guests that had like cranial windows, and basically
that's crazy.
you have access to the brain tissue, and you can see with specific instruments like the 2-photon microscope, for example. And it's very interesting. But as you said, in vitro, you cannot recapitulate all the complexities of this kind of 3D development because, as you guys know, the brain is 3D. We live in a 3D world, so you need to have this complexity. And also you don't have blood cells, you don't have this kind of thing. So it's really, really important to have also studies that have been done in living animals in order to really address a problem on its core and not only like brush the surface, so to say, right?
Yeah, so that just to, maybe kind of chime in here, when we talk about, when my students and I talk about development, we say that it's 4D actually, because the 3D component, of course, you have stem cells going, but then there's also time. That is a very important determinant of the output of our progenitors. And also, I don't want to by any means invalidate in vitro research. I think it's a very powerful way to address a lot of these things. In fact, my first paper used a lot of in vitro systems, simply because for some developmental questions, this is actually a better system because there's situation is crowded in vivo, and in vitro you can really distinctly characterize whatever you're after. In our case, it was the acquisition of axons and dendrites. And for that, actually, plating or putting the dissociated brain tissue on a petri dish allows you to really recapitulate the milestones of development that you also see in vivo. So, yes,
that's totally correct. I completely agree with this. I myself study a lot of things in vitro because it's just there are no tools for in vivo, and it's also super hard to approach this problem. And as you know, we are not allowed to do in vivo experiments in Germany anymore.
Yeah, we are not. No, well,
it's coming. But this is another topic.
We're trying to. Yeah, we're trying to. It doesn't come. Yes.
Here, I would like to refer to another podcast with Roman, who we talked about animal experimentation. If you guys want to know more, just go and listen to that one. All right. So cells in vitro, in vivo. Yeah. But your research also has another aspect, and this is like a little bit deeper than the cell, and we go to the molecules, etc. So let's first define what do we mean by the proteasome, and what is this thing? Why is it important in brain development, for example?
Yeah. So that's a question that we kind of want to understand. So we from a textbook knowledge, what I always say when I'm talking about these things is that in the 70s, Crick has published a paper in Nature defining the dogma of molecular biology, right? And then he said that there's a DNA, and then they understood by then that the DNA gets transcribed into a message called RNA, and that message then gets translated into a protein chain, into a polypeptide chain called a protein. And these molecules are the ultimate kind of product of DNA locus, right, of a gene locus. And so a lot of the studies that has happened in the last 10 years have addressed the identities of cells just based on their RNA. And this is because of the technology. So this is now possible. That's why we are using it. However, for reasons that I think we are also going to go in later, in the brain, it's a little bit more complicated than that, because neurons not always want to use that message, So for us, the studies of proteins is really what is quite interesting and what is inspiring and drives us. So what was the question? Was about the proteasome. I see. So like everything, in biology, it has a synthesis phase and it has a degradation phase, right? And it's no different than no different for protein. Proteins get synthesized at the ribosome, right? And then they get degraded at the proteasome. So it's a gigantic enzymatic complex, and it allows to recycle amino acids back to the cytoplasmic pool to enable more proteins to be made where they are needed. Also, I was just at the conference at an EMBO meeting in Croatia on ubiquitin and proteasome in health and disease, and there was a fantastic talk by Brenda Schulman, and then she said, well, proteasomes are great because they degrade proteins, and if the proteins are misfolded or if the proteins are broken, then you're not only recycling the amino acids, which are sometimes coming from an environment and it's difficult to get them, but you're also protecting yourself from the exposure to aggregates. However, it costs energy. So for a 50 kilodalton protein, you need 70 ADP molecules, which is really remarkable. How there's this molecular currency as well, right? For the synthesis of the protein, but also for its degradation. So this is something that kind of is baffling to me, right? Because normally when we take out the trash, you also have to pay. But yeah, maybe that's just me thinking. All right.
Well, it is maybe our Eastern Europe upbringing where
maybe that
Western Europe
we just throw it in.
It's different.
But now in Germany, so
no, but it's also not only like synthesis and degradation, but you also have trafficking, right? So
yes,
you still have a little bit more energy, and ATP is the universal currency in biology, if you wish, the energy. We measure things in ATP. How many ATP do you need to do this and that? And it's very interesting. But neurons are also fascinating because they're like, once they are differentiated into neurons, when they become neurons, they stop dividing, right? It's called post-mitotic cells. They live basically our whole life, right? And you have immense amount of protein turnover, right? So it's important for the cell to keep track of these proteins that are synthesized, that are degraded, and then have this kind of a balance between energy and et cetera, et cetera. You mentioned two terms. One of them was protein aggregation. So let us differentiate between things that are happening in the development and maybe in the later stages when we are already getting old, etc. For example, with aging. Yes. So do you have protein aggregation in development or no?
I think you do. It's something that we don't really do in the lab, but I think this is a process that also happens early on. It's just I think that a cell is more fit to deal with it early on rather than in comparison to later. We look at accumulation mostly because of the defects in the degradation pathways. So, but this is. It's probably even known. It's embarrassing a little bit, but I don't really know. I have to admit. So, yeah.
Okay. Maybe we should look up.
We should definitely look up that.
Yeah. But how do you know that you're dealing with this kind of ubiquitination?
Yes.
Right. What is this process?
Right. So then this proteasome it knows what to degrade because it has this filter that the cell has developed very efficiently and very smartly. And it's called a ubiquitin molecule. It's a small protein that gets conjugated to target proteins that then are sent off to the proteasome for that degradation. So a lot of people call ubiquitin like a kiss of death, for proteins, because once it's on the target, it is recruited to the proteasome and gone. Ubiquitin is then recycled. Now we know the situation is far more complicated than this. So there's ubiquitination that doesn't do it, and there's this non-canonical function of ubiquitin. Now we know ubiquitin is also conjugated to non-proteinaceous matter. So we have nucleic acid ubiquitination, and people just start to understand. First of all, start to see it, and second of all, start to understand what it does. There's fantastic work from many different labs addressing this, also in disease context. But essentially, ubiquitin is my student, Nina Koch. She pitched a term called, so she said that ubiquitin acts as a molecular Tinder because it brings, target proteins to the proteasome, so acting as a matchmaker. But then, I was giving a talk and I kind of recalled that, and then I realized, well, it's not really nice because it doesn't end well, right? It doesn't. It's not a happily ever after scenario because then the protein gets degraded. Hopefully, right?
Well, we have a lot of Tinder dates that also.
We definitely, for sure.
I don't know. I've never had Tinder.
I'm hoping nobody gets degraded. Then, but
this is the thing. Yeah. But why is it so important to have this kind of you mentioned, right, degradation systems to keep the balance, but what happens when one of these machinery it breaks down, for example?
Yeah. So this is what we look at. So there is, I have to say, the field of development and the field of ubiquitination, they are not yet talking to each other. Now and that's why we're very excited about this, because we're very interested to look at the developmental context of a lot of modus operandi of these molecules, right? And so ubiquitin gets conjugated to a protein in a very complicated process. Well, not very complicated, but it's a cascade reaction that involves enzymes of different classes, and ATP, as you mentioned, and magnesium, and then there's but essentially the last step of that reaction is catalyzed by enzymes, so by proteins that have a kind of a, I don't even know how you would define an enzyme in that friendly way, right? It's a protein that that enables a reaction, right? So
speeds up the reaction.
Speeds up the reaction. Yeah, catalyzes the reaction. Genau. So we would have proteins that are called E3 ubiquitin ligases. So again, like an unfriendly name, but we call them E3s just for simplicity. And these E3s recognize the protein that needs to be tagged by ubiquitin, and ubiquitin itself brings them together and links the two. And so it happens that we, in the human genome, have over 600 of these enzymes, of these E3s. And they have obviously quite a spectrum of specificities. So each ligase would not only see one protein, but many. Some see a lot, some don't, right? And it is fascinating that if you look at the genetic connection between the E3 genes, so the connection between E3 genes and human disease, we see around 15% of all these E3 genes are directly linked to the neurological disease, which to me is really mind-blowing. But it also demonstrates that for some reason, development or neurons, or both, neural development, really depends on that system quite heavily, right, for it to work well. Otherwise, there would not be the 15. I mean, 15% is a lot in biology, right? And basically, what happens in a lot of these scenarios, and they can be different scenarios that, mostly genetic mutations in E3 genes. I'm saying genetics like 40 times, but the substitution. So they lead to loss of function of E3s. So then the protein that was supposed to be degraded now is not, because the E3 cannot bring the ubiquitin on it. So it doesn't have a way to catalyze that conjugation. And that in turn, so if that ubiquitination was of canonical type and it was supposed to end up at the proteasome, that causes an accumulation of targets of that given E3s. And this is kind of what also my lab has been doing for many years now, is a hallmark of some of the neurodevelopmental disorder, the accumulation of substrates that rely on the degradation pathway that is mediated by the E3.
Which diseases, if you may say?
So, my lab particularly studies the developmental scenarios in autism spectrum diseases, for example, or intellectual disabilities. This is a broad heterogeneous group of disorders. Quite now in the realm of modern genetics, we can narrow down the disease type. And one of them, for example, that we have published in 2021 was Kaufman oculocerebrofacial syndrome, so KOS, K-O-S. And this is a rare genetic condition or disease that affects kids, and it affects them quite profoundly. So there's developmental delay, there's autism-like features behaviorally, but there's really dramatic kind of brain image where the connection between the left and right hemisphere is very thin. The ventricle in the brain is dilated. There is obstruction to the cerebrospinal fluid flow through the foramen magnum. So there's a lot of kind of structural issues in the brain of the kid. So there, what we have done is, we have, and the cause for that disease is the mutation in one of the E3s. The gene is called UBE3B, and what we have done there is really, this was one of my PhD projects, and we have really chased down what UB3B ubiquitinates in the brain and where, and then kind of narrowed down the cascade that might be associated with that disease in humans.
That's fascinating. That is fascinating. Do you know at what stage of the development, of neuronal development, stem cell development, the mutation occurs? The mutation. So this is also something that we, together with my dear collaborator here from Göttingen, Sonia Lawrence, we just wrote a little perspective article on this, and we have really made sure that we mention the spatiotemporal aspect of the ubiquitination signaling, and. So I think one of the messages from that article is that it actually does not matter so much, when the mutation happens. It matters when the ligase is actually needed, because a lot of these diseases would stem from, the pivotal role of your ligase in stem cells, but some of them actually act only later at the synaptogenesis, right? And in the case of UB3B, it was indeed
the UB3B seems to be necessary not so much for the stem cell progression towards a neuron, but for an early neuronal development. Since we have found that when we look at the knockout neurons in our mouse model system, we have found quite simplified dendritic arbor. So these are the neurons looked very, the neurons are very simple, right? So normally you have this crazy, beautiful branch. They look like trees, and you have many different points of contact between them. In this case, the dendrites were very, very simple. They had more synapses. So the phenotype was indeed quite complex, but it was really indicating that UB3B was necessary for this, early kind of events in a maturation of a neuron rather than.
It's just fascinating that in a cell you have
It is
hundreds of thousands of proteins or molecules running around. It's so densely packed, and then you kind of break down only one, and then there is a complete phenotype. You have this disease all your life, and that's it. You cannot do anything about it. It's just incredible, and we still struggle to understand why, what was it? Is there any idea why is this happening besides mutation? But can you reverse it? Is it possible?
So this is kind of what the paper gave a perspective on, in a way. We have found that but then again, this is a very defined molecular and cellular scenario, right? It's not the case for it's not a recipe. I mean, we would like it to be, but it's not yet there. So in the case of UB3B, we have indeed found that so the ligase operates locally, in fact, and it operates at the synapse, in fact, and it is supposed to, in a normal condition, degrade a large phosphatase enzyme, and so that's how the paper finished. We have also done behavior experiments. We have done electrophysiological experiments. We have really characterized the knockout quite well. But we finish with that substrate being, we say, okay, it's locally and it's at the synapse, and in the knockout it accumulates. So then I had a couple of students that came in Berlin to follow up on this, and we tried to ask. So now, knowing that there is accumulation of an enzyme again, that is a phosphatase, can we now target the phosphatase activity to ameliorate some of the behavioral phenotypes that we are seeing in the UB3B knockout mice? But we tried. But I don't really know. There needs to be more work done there for sure. But I think what is really my main motivation also, and I believe my team as well, is that you don't really work towards a drug. We just can't do it, right? And you need really a company for this, or a gigantic department. But we open these avenues that we show where you could maybe potentially ask another question in an animal model again to see what you can do. And I think it's quite, I don't know, I may be biased, but I think it's, in the case of ubiquitination, when you're dealing with accumulation and more, I think it's a easier scenario than when you're dealing with something like a mutation that causes less, because it's difficult to induce. But now with the drugs, mostly inhibitors, you can easily control the activity, right?
That is correct. Very interesting. This is called basic science, right? Basic research.
It is basic. It is it's a basic research rooted in scientific excellence, but with a kind of a segue into maybe clinical applications, for sure. Motivated by maybe like this.
But it's cool that it actually has the clinical potential, right? And a lot of studies that have been performed, in many papers you read. And this, we might cure that, and we might cure it, which is completely, it's nonsense, but it's packaging the paper and you sell it better, right? We can maybe talk about it a little bit later in our second part of
No, I think people shy from saying that it's a basic science, and there's nothing bad with that, right? If it's excellent, then it's nothing bad with that. But you're right.
We are funded to do this. In this case, right? I don't know. I remember studying development, neurodevelopment. For me, it was really I was not familiar at all with this field, but it's very interesting, but it was a little bit too complicated for me. I stepped back and I went to synapses and memory and things that I'm most passionate about it.
Yeah, but that's the thing, if you meet people at the Congress and it's a developmental neuroscience session, I don't know what that means, because development is everything.
That's
what
correct.
you just described, synapses and memory, this is also development,
Correct. Totally
so
correct.
development is everywhere. Some people even say that neurodegeneration is neurodevelopment, in fact, right? Aging is neurodevelopment. So, I mean, you really have to make sure what you're defining. In my case, it is the OG embryonic development of the brain.
This is very distinct.
Very distinct, yes, exactly.
Yeah. I'm interested in things that are happening in later
on, yes.
But it's still fascinating. I mean, all of the topics are so incredible and so complex, and I don't know. It's one of these fields that you can maybe work 3-4 lifetimes and still be surprised with what is happening.
I don't know. if I could again, maybe I would go into synaptogenesis, and synapses. And
who knows,
right? Yeah.
With all the technology and all this interconnectedness that is going on around the world,
now,
it's just crazy. You can literally, we have like a collaborator that agreed to inject animals with our viruses in a short notice, and I will go soon to another country, explore this kind of experiments, because it's challenging to do these things on a short notice in some countries. You mentioned the organoids models, right? Do you have them in your lab?
So we collaborate a lot in the lab, and really, actually, I really also believe from early days. This is maybe because of my mentors, but I really believe in the power of collaboration. As you said, you can't know it all, you can't do it all, and some people just do it so much better, and it's just fun to learn. Always learn, never stop. So we collaborated now on a current story that I am supposed to work on right now to package it into a paper and submit. It's a story from one of the first PhD students from my lab, Katie Cuthill, and my fantastic postdoc Katya Borisova. And it's a study where we also went to the developmental disease, to the same route as we went to UB3B. We looked at the homolog of UB3B called UB3C, shockingly. It also is associated with a very severe neurodevelopmental disorder. And in Berlin, there's a lot of this kind of alternative models to replace and reduce animal research. And I met an incredible collaborator, Agnieszka Rybak-Wolf, who is now a core facility head for organoid development at the BIMSB Institute, was on a part of the MDC in Berlin, and she has done these experiments for us. So we have. I can't say that I have them in the lab, but we are definitely working with them. So we collaborated on that project with Agnieszka, but. My student that I mentioned, the molecular Tinder student, she's now in Paris for an internship, and she's looking at organoids for another, disease. And so I think it's a new model that I think it's polarizing. I have to say, I don't know if it's polarizing still. I think nowadays you can build a lab that is extremely successful that just uses organoids, and we've seen incredible work that has been published. We have seen quite groundbreaking data on that, because it does allow you to look into a more human related context, and as I said, there are fundamental differences between the way stem cells produce neurons in a mouse and in a human brain. It's a model, and I think that as is with living animals, living mice, as is with infants that you put in the MRI machine, every model, if you will, I don't know if this is right now to say that infants are model, but your biological kind of model, every model has limitations and has advantages, right? And I think it is also the case for brain organoids. I think for what we are doing, they're really helpful. Then that's probably what I want to say about that.
Yeah, they're fascinating. I mean, really, you can address problems that happen in a very early development, and this is not possible. Not possible, but very, very, very hard to do it in other model organisms, and in some even not possible. I remember one of the very first papers from I think Madeleine Lancaster, one of the pioneers in this field, was it on microcephaly? I don't remember. One of these diseases that they could not even study in mouse, but they found in some mechanisms about this disease in this model, which is incredible, right? It's crazy. We have also a podcast about this. I think the second episode. So go ahead and listen to it. Collaboration, very important in research.
Yes.
And you are part of one very interesting network.
Yes.
It is called FENS-Kavli Network of Excellence.
Yes,
right?
correct.
Can you tell us more about this?
Yes. Yes. How exciting. So as I said, the collaboration has always been in the core of my work, just because I as I said, like there's so many fascinating scientists and incredible people that can teach you so many useful and interesting things, also unuseful, but it's still interesting. So the network has been, I have been a scholar for I think a year now. I have been inducted in 2023 in September, and it's I've loved it since the beginning. It's a network of 30 early to mid career neuroscientists from Europe based.
Can I interrupt you here? Can you define early to mid career?
No,
Okay.
Actually, I don't know what that means. I think you can define it. I mean, I really don't like when people do this, but I don't even know what that means. I think I think the point is, maybe it also reflects kind of a spirit that you have, and you're not established. You're not you're not a full professor with a tenure, because I think the problems are quite different, right? Or the problems or the advantages. So the network is composed of 30 researchers. So the mandate is for 4 years in the network, and every 2 years the 15 scholars rotate, and new are selected, and the 15 go into alumni. And the network is composed of these two core branches, the FENS and the Kavli. And FENS is obviously the biggest European organization that advocates for neuroscience, for excellence, for scientific exchange. And we have the Kavli Foundation, who does that in the States, right? So they also promote scientific progress and excellence, and also outreach to the public through a lot of initiatives globally, really. Yeah.
So how do you become a member of this network?
So they have calls for to apply. I think what really is the common denominator for the scholars is this. Scientific excellence is definitely one of the must, but I think you'd imagine. I want to imagine that every scientist that works in Europe wants to be excellent. You just have to demonstrate that you have managed that, and you need to be really passionate about science, not only in terms of your conduct in the lab, but also what that means outside. So what the network really does is incredible work in advocacy for brain sciences funding at the level of European Parliament. We also do a lot of outreach to the outside. So we do a lot of community engagement, but also quite a lot of events at FENS. So the FENS conference that is biannual. There's also a lot of stuff on mentorship, so mentoring. There's a lot of prizes. There's PhD prize. There's mentoring prize. There's the joint initiative with Alba Network, which is promoting diversity in neuroscience. So there's quite a lot. And I think what I really appreciated about the network is really, if you're passionate about something, you can make it your agenda. And they, if it's really not against, I would say common sense, then you will find support for doing what really is close to your heart.
So, a very tough question: what did you give to this organization, and what did you gain so far?
Okay. Tough question. No, not really. I think you give time. You give your time for sure. You want to jump in on a lot of initiatives, and I did. A lot of these things, I think I can support a lot, but there are people that are really professional about how they approach advocacy. For example, we have like in a committee, a special committee that makes sure that this is professional and well done. I think what I gained was, really on a personal level, to be surrounded by people that are so inspired to change something in Europe in terms of brain sciences, neurosciences, and science conduct in general. To be surrounded by people with this spark, and that energy that, it's really sometimes a numbers game. So as a group, we can really do something together to support people. We have now done in Vienna last year, where we also met. Laura Sweeney and Sandra Siegrist from ISTA have organized. They were organizing a young PI symposium, and I had the pleasure to be on a co-organizing committee. And it was fantastic. It was an event that brought young PIs, really young. So, I think there was a definition too. So you're supposed to be like 8 years after a PG or 2 years after starting from your group, or I think it was a transition. Actually, sorry, this was a transitions to independence symposium. So you could be a PI, but you can be like an advanced postdoc that is thinking about starting your own group. And this was a symposium where what I really appreciated is you can, for a day, really talk about problems that you face in the lab and realize it's not only you who's facing these issues, and you can exchange that and maybe together come up with a solution. But there was also a lot of positive things that we focus on, which is scientific exchanges. So there was a lot of collaborations that were sparked in that event. And I mean, this is really invaluable. I think that, you can read a lot of papers to be a great scientist. You can spend a lot of time in the lab to be a great scientist. But if you don't have a sparring partner, or if you don't have a common goal, like what I think all the scholars in FENS-Kavli have, then you're only gonna go there this far,
Okay, I totally agree.
Yes.
I was a little bit surprised, to be honest. I come to Göttingen, and there is this program that is amazing, etc. And
Yes.
you have other programs and a lot of excellence initiative going on. But still, like the amount of students that really want to do something like outside of their daily routine is not that high. Yeah. And it's when you start some of these initiatives, it's a little bit hard to do it by yourself. But
Exactly.
Always I see here and there some sparks of motivation, ambition, etc. And therefore, I think that indeed the network and the value to have contacts to people who are like-minded like you, and they would like to contribute to the community, to society, if you wish. It's really important, and it drives you, and then it drives them back, and it's kind of a mutual push system.
Absolutely. And then for example, I mean, this really has a domino effect, or like I don't know, maybe even a butterfly effect. It also affects the people in your lab, right? Because you also give them the chance to be exposed to a different side. For example, now they had scientific exchanges for scholars, and my postdoc Katya was visiting the lab of Georgia Rapti at EMBL, and she learned how to work with C. elegans. This is something that we haven't ever done in the lab, because we were always quite mice centric. But she had that incredible once in a lifetime really experience because of that because of that exposure, because of the network. What network has really kind of given me. So I mean, I think that these things are really invaluable if you think prospectively.
Yeah. You're also involved in another activity. You kind of mentor young early career scientists, give them advices. What would you say to a researcher who wants to start their group, for example, right now? What would be like let's say 2-3 key messages?
So it's a job that is quite peculiar, right? I don't even know if you can say that it's a job. It really is a lifestyle that you're choosing. But when they are already thinking about starting a group, then they know that already, right? So we don't have to say that. I think what was, and then I think there's as many. I think the question can be answered in so many different ways, and I think I can only come from my own experience. And I think I have had incredible mentors along my scientific kind of journey, So, and I think without the mentors and without that spark of collaboration and kind of like, bouncing ideas off of each other, I don't think I would have stayed, because this is really something that you need to feel supported. You need to have someone in your corner. And, for me, this has been really instrumental. So I would say that this is very important. Second thing, what I really believe in is the power of creativity. And I think that this maybe is also not really addressed, but for that, you really need to have a great environment. And by that, I mean, peace. I think we scientists, we appreciate when people leave us alone, right? So you need to really sit sometimes for hours and think, like in the Google creative scheme, it's like 20% of time you're supposed to be thinking and not doing anything else. So you need an atmosphere, you need an institution which allows you, for some sort of a scientific freedom. Of course, I'm not saying that, you shouldn't contribute to the faculty jobs, or you should, of course. But what I'm saying is surround yourself with like-minded people, have good mentors. And I think what is also very important is this exchange that we mentioned. And some people call it collaboration, but I do think that exchanging your own ideas and, talking to your peers, talking to people that are senior to you, don't become them, but talk to them, maintain your identity. And if it's not your dream, don't do it. It has to be your dream, because you have to make sacrifices. And that's probably what I would say right now. But ask me in a year, I'll probably change my mind on something.
Yeah, I mean, these are valid things, right? And also sacrifice, I think it counts for every profession.
Every profession, really, right? I mean, what do we? Sacrifice. I mean, what does it even mean, right?
Yeah. It's if you want to be the best or whatever, you have to sacrifice time with your friends, family, social life, etc. And concentrate, deep work. We've been there. Yeah. Probably we are still there. I don't know.
Yeah, for sure.
Who knows? One last question before we conclude our first part of discussion. I'd like to ask you the following. So imagine you have unlimited amount of financial resources and you are in charge of leading a scientific institution for 10, 15 years, for example. Mm-hmm. What would this institution look like?
Oh my God, how exciting. Oh my God. Okay. So first thing, what really has been really, for me, also career changing in a way, is exposing myself to other science that I don't really fully understand. And that's what I would do. And I think that this is it's not an original idea, right? There are places like this, Ingenielia is like this, or Max Planck actually, by organizing it in a society way. But I would, my collaboration with Sonia Lawrence on structural biology, my collaboration with Matthew Crouchar on some of the translational processes that happen in the brain, these have really shaped my science and the way I understand and the way I think about molecular level, cellular level, systemic level, organismal level. So I think if you bring people in the same, and this is kind of also echoing on the FENS-Kavli network, because if you bring people together in one room with different type of expertise, we have like cognitive neuroscientists, we have behavioral scientists that are interested in animal behavior, we have people that are interested in mitophagy, or people that are interested in helices and cryo-EM, and then people that are interested in activity dependence. Bring them in a room, give them like an hour, and you'll just see scientific dreams really come true. And it's just so exciting to work this way, and with people that are also striving to learn more. And I think so if I had an unlimited amount of money, then that's what I would do. Mm-hmm. I would just bring people from different parts of neuroscience and also from outside of neuroscience, right, to join the conversation. Mm-hmm. So you can tackle the mysteries of the brain.
Mm-hmm. And this would be done somewhere in the world? Is there a particular place that you wish to do it?
Oh my God. Okay. So now we're talking geography. I don't know. Yeah. It has to be a place that respects the scientific, no boundaries. Okay. Mm-hmm. So it has to be a place that respects the needs of scientists. Mm-hmm. So then, probably, I don't know. This is a tough question. This is a tough question. It should be probably a nice location. A lot of scientists go running. It should probably be close to the beach,
or the mountains,
or maybe both. Like Madeira is very nice for that. Like the island of Madeira. I don't know.
A lot of distraction. Sorry. Oh my God. I mean, you will be distracted by the beach or whatever, or the nice nature.
Okay. Well, then, okay.
Maybe not.
Remote island somewhere, so that I don't know. It's a tough. I think geography is a tough question, but yeah.
Yeah, it's an interesting, right? And I have a dream. You know, as Martin Luther once said. I think we can make a lot better science. We can do a lot better science. We can build our institutions in a different way and to promote this kind of activity. And we can also perform better in education if you wish. So for me, like the dream is to kind of be involved or like initiate some kind of institution that is combining a research institute with a company and a university-wise, kind of you have the circle, you study, people teach you from both aspects. Then you have things that, something like, Bell Labs was not like this, but it's a very interesting example when you have a lab that created so much innovation. They have 8 Nobel Prize winning people there or something. How do you drive this kind of creativity?
I think for me also the problem was that I always struggle with this. How you're a kid and you're supposed to know already what you're going to do as an adult, but then translate this into science scenario. You're a student that is in the program, in the neuroscience program, and now you're supposed to decide where you're going to spend the next 5 years of your life doing PhD. Without rotations, which are also taking forever, and you can only have 3. How do you even decide on that? So I think maybe what you're saying is very not maybe, but it's very valid. Like if you create incentives for people to be exposed to a lot of different things, then it also makes them more conscious of their own choices. And their dreams become a little bit more tangible and formed, because I think a lot of chaos also comes from lack of direction, right?
And you have a lot of information nowadays, which is unstructured and everything, and then it's crazy.
Yeah, exactly.
It's crazy out there.
Exactly.
Well, what should I say? Let's hope for the best, and maybe, in 10, 20 years, we can have a very nice institute.
So Island Beach. So wait,
Island Beach.
That's great.
A lot of margaritas and a lot of science.
A lot of cryo-EM and also behavior and also like cognitive neuroscientists. Yeah, let's. I love it.
Who knows? Yes. Life is unpredictable.
It is.
Right. Mateusz, thank you very much for coming from Berlin to Göttingen for our conversation. And with this, I would like to conclude our first part.
Thank you very much for having me. It was a pleasure to talk to you.